Hi, my name is Harsha Sirigireddy.
I'm Sami Marchand. And I'm Jade Bates. There's a great chance that you or someone you love will be affected by cancer within your lifetime. Currently, the average survival rate amongst all types of cancer is only 68%. But what if we could change that number? New research in quantum dot technology may be able to do just that. These tiny man-made structures are helping to revolutionize bioimaging and increase early detection of cancerous tumors.
Before we get into bioimaging, we will talk about fluorescence, the mechanism by which quantum dots function. Then, discuss the role of quantum dots in bioimaging. And finally, how quantum dots have become more sustainable.
Fluorescence. The earliest known examples of the phenomenon known as fluorescence was observed in nature. For example, coral may contribute to photosynthesis by converting unusable wavelengths of light into ones which can be used by algae. Another well-studied example of fluorescence is an organism known as Hydrozone Aquaria Victoria. This jellyfish lives in the photic zone off the west coast of North America and is identified as a carrier of green fluorescence protein, also known as GFP. The gene for green fluorescence protein has been isolated, and this is significant because it is used in a lot of genetic studies. But since then, we found alternatives to fluorescent proteins. We've been able to synthesize organic molecules and create crystals that have fluorescent properties.
To understand how fluorescence works, let's look at a simple diagram. This is what's known as the Jablonski diagram. When a molecule absorbs light, it goes from a ground state to an excited state. When it returns to the ground state, some of that energy is lost due to vibrations. The rest of it is emitted as light. This is known as fluorescence.
Fluorescence has many commercial applications. You have fluorescent lamps, you have a fluorometer, which can be used to detect very low concentrations of gas. You can even find fluorescent pets. But the most important application related to our topic is bioimaging.
Before we can get into bioimaging, let's talk about the crystals that Harsha just mentioned. These crystals are commonly referred to as quantum dots. Quantum dots are man-made, fluorescent, semiconductor nanocrystals. Typically, quantum dots are made of binary compounds such as lead sulfide, cadmium sulfide, and indium arsenide. The crystals are made with different diameters depending on what they will be used for. The diameter of the crystal will control which color of the light spectrum that will be emitted. The larger the quantum dot, the more energy that is absorbed, resulting in a higher wavelength of light. A smaller quantum dot will have a smaller shift in energy and in wavelength. So as you can see, the 2 nanometer quantum dot emits blue light, while the 6 nanometer quantum dot emits red light. So, how do these tiny dots have the potential to save lives? Quantum dots can be used for medical imaging, such as the detection of small cancerous tumors. Current imaging techniques depend on organic dyes with a broad spectral width, which limits the amount of colors that can be emitted. They also have shorter lifetimes, which decreases the time the needed tissue can be tracked. Quantum dots' small size makes it easy for them to travel anywhere in the body. They have a better resolution, they can emit the whole light spectrum, and they have a slower degradation time than current imaging techniques. If quantum dots are so great, why aren't we using them now? The answer is simple. The materials quantum dots are made of are too toxic to be injected directly into the body. What's the good in early detection of cancer if the mechanism used to detect it could kill you? I will now hand it over to Jade to describe the new technology that would make the quantum dots more sustainable to human health and possibly lead to use in the medical field.
As Sami mentioned, the toxicity of quantum dots has limited their ability to be used for biocompatible imaging and other biomedical applications. However, what you see here is a simplified illustration demonstrating how a group of scientists from the Bio-Nano Electronics Research Center in Japan are changing that. For the first time, manganese-doped zinc sulfide quantum dots which you see all the way to the left, have been stabilized by biologically conjugating them with marin, an extremophilic bacterial polysaccharide you can see to the right of the unconjugated quantum dots. The hybrid quantum dot marin structures are less toxic and therefore more sustainable. To obtain the hybrid structures, the unconjugated quantum dots are suspended in a solution prepared by mixing powdered marin with water, then centrifuging the solution to obtain a pellet-like product. The graph seen here shows the results of cytotoxicity studies done on mouse connective tissue cells using increasing concentrations of unconjugated or bare quantum dots, hybrid quantum dot marin structures, and bare marin structures. The unconjugated bare quantum dots are indicated with red, the hybrid quantum dot marin structures are indicated with blue, and the bare marin structures are indicated with green. The hybrid structures increase cell vitality by over 200% in the lowest concentrations and up to about 500% in the highest concentrations compared to bare quantum dot results. This development of biocompatible fluorescent nanoparticles introduces the possibility of breakthroughs in bioimaging, which could improve detection, diagnosis, and even treatment of cancerous tumors.